Hello everyone. Today we'll be solving Cambridge International AS and A-Level Biology Paper 4 A-Level Structured Question. October-November 2021, Variant 4.2. Today we'll be solving this particular question paper from question number 1 to question number 5. Question number 1A. The Bowman's capsule of a nephron is involved in ultrafiltration. Figure 1.1 is a diagram of a part of a Bowman's capsule and glomerulus. So we can see, we can see the capillary wall. A is basically the capillary endothelium cells. And we can say that this is B is capillary lumen. Alright. And C is basement membrane. D is basically podocyte cell. E will then be obviously glomerular filtrate. And F is basically the Bowman's capsule. So the first thing that they're asking, the feature glomerular filtrate. So glomerular filtrate is E, basement membrane is C, podocyte cell is represented with D, and capillary endothelium cell is represented with A. B. Describe and explain how the structures in the Bowman's capsule and its associated blood supply are adapted to allow ultrafiltration to take place. We know that the efferent arteriole is wider than the efferent arteriole, and this causes high blood pressure in the glomerulus. In the glomerulus, there are pores in capillary endothelium which allows filtrate to pass through. The basement membrane, which is located between the capillary endothelium and podocytes, acts as filter. The podocytes have long finger-like structures called the foot. It holds the basement membrane in place. Basement membrane stops large protein and molecules above relative molecular mass of 68,000. Alright, it doesn't allow it to pass through, and allows glucose, amino acid, ions, and water to freely pass through. Part C. The glomerular filtrate rate, or GFR, is the rate at which blood plasma is filtered in the Bowman's capsule. Figure 1.2 shows the relationship between GFR and mean renal arterial blood pressure. The mean renal arterial blood pressure, as it increases, the GFR, at first it increases from 5 to 10 kPa, which then increases up until 100, and then it plateaus up until 20, and then it increases again. Comment on the relationship between GFR and mean renal arterial blood pressure. So as the mean arterial blood pressure increases, we can say as it increases, alright, GFR rate increases and then levels off and then increases again. The plateau occurs at 100 cm3 per minute between 10 and 20 kilopascal. The plateau is normal, healthy value. Part 2. Suggest one reason why GFR of a person might decrease. Well, the GFR might decrease due to dehydration. Also, if the person has low blood pressure. And followed by that, if the person has kidney damage, the GFR can also decrease for that. Question number 2. Scientists are researching new ways to reduce the global atmospheric carbon dioxide concentration. There are concerns that an increasing atmospheric CO2 concentration may lead to effect that decrease biodiversity. Give one example of human activity other than deforestation that contributes greatly to the increase in global atmospheric carbon dioxide concentration. So basically any point such as industrialization or like transportation, power stations, burning of fuels, or like home heating, burning of fuels of any sort. Algae are aquatic photosynthetic protocrists. Some researchers genetically modified the unicellular algae Chlorella vulgaris to try to increase the rate of light independent stage of photosynthesis. C. vulgaris was modified to increase the expression of the gene coding for the aldolase. Aldolase is an enzyme that causes an increase in the concentration of rubisco. Okay. Two cultures of C. vulgaris, one that was not genetically modified and one genetically modified, were grown under controlled condition for 14 days. Samples were taken from the cultures at regular intervals during 14 days to obtain measurements of dry mass. The results are shown in figure 2.1. Unmodified and genetically modified. We can see genetically modified has a higher dry mass throughout the experiment compared to that of the unmodified one. With reference to figure 2.1, describe the difference between the results for the two cultures. Well, they started off at the same level, but then they have a different level of biomass. Genetically modified C. vulgaris have higher dry mass throughout the experiment. Genetically modified C. vulgaris has slightly higher rate of increase in dry mass, as we can see. There is a slightly higher rate, in 14 days, where it went from 1.2 to 1.5, and then C. vulgaris was in like 1.75, 1.8, similar, somewhere there. All right. The largest difference in dry mass is after 8 days, as we can see. There is the largest difference. At day 2, the difference between two C. vulgaris is 0.09 gram per dm cube. Day 2. All right. And at day 10, the difference is about 0.28 gram per dm cube. Question C. Explain how the Calvin cycle was affected by the genetic modification of C. vulgaris. Genetic modification increased more rubisco, so more carbon dioxide fixation can occur. As a result, more GP is produced. More GP then can be converted into more TP, and finally, more regeneration of RUBP is possible. So, greater rate of Calvin cycle is possible. Carbon fixation is a rate limiting step, as the concentration of rubisco is a limiting factor in carbon dioxide fixation. Intermediate products of the Calvin cycle are needed to produce organic molecules for use by the cell. Describe how these organic molecules are used by the cells. So basically, glucose is used for respiration, starch is used for storage, cellulose is used to make cell wall, and sucrose is used for translocation. Fatty acid and glycerol is used to make cell membranes. Amino acid is used for making protein, and protein is needed for growth and repair of cells. Part E. Planting large number of trees is one way to reduce global atmospheric CO2 concentration. Large-scale culture of genetically modified C. vulgaris could also reduce global atmospheric CO2 concentration. Suggest one advantage of using genetically modified C. vulgaris instead of trees to reduce global atmospheric CO2 concentration. So basically, C. vulgaris have a faster growth rate, and it is cheaper to set up production of C. vulgaris. C. vulgaris is aquatic, so it does not use any land which can be used by other crops that are grown in land. Question number 3. During the germination of barley seeds, amylase is produced. Describe the sequence of events that leads to the production of amylase during germination of barley seeds. So at first what happens is that the barley seeds embryo absorbs the water, and this stimulates the embryo to produce gibberellin. The gibberellin then moves into the alluron layer of the seed, and then the gibberellin binds to the receptor, and the receptor then binds to the enzyme that breaks down the delaprotein. Now, delaprotein was preventing the transcription factor PIF, all right, from binding to the amylase gene. So once the delaprotein is gone, the PIF can now bind with the amylase gene. The gene coding for amylase is then expressed, and the translocation of mRNA and the translation of mRNA of amylase enzyme produces the enzyme amylase. Part B. Malting is a process involved in the production of a drink called beer. During malting, barley seed germination is controlled so that the sugars produced during germination can be used in the production of beer. Figure 3.1 shows two features of a germinating barley seeds during the first 5 days of malting. Activity of amylase enzyme, the percentage of starch reserves remaining in the barley seed. So the starch reserve, we can see the starch reserve is going from 100 to 85, 80, 82, 100 to 82. All right, and then we can see the activity of amylase in arbitrary units is going from 0 to day 5 at 96%. So 96. Amylase activity in arbitrary unit is
8.4.
State the precise location of the starch reserves in the barley seed. So the barley seed contains starch, and then the starch is mainly stored in endosperm. So endosperm will be the answer. Part 2. With reference to figure 3.1, describe and explain the effect of malting on amylase activity and the percentage of starch reserves remaining in the germinating barley seed. So we'll have to describe and then explain. So amylase activity increases from day 0 to day 5. We can see that from day 0 to day 5 it is increasing. All right, so as the seed is producing more and more amylase, and as a result the percentage of starch also decreases because they're getting more and more hydrolyzed. At day 0 the amylase activity was 0 arbitrary unit, and at day 5 the amylase activity was 8.4 arbitrary unit, and 82% of the starch was remaining at day 5. Part C. In the malting process, germination is stopped before the concentration of sugar in the germinating barley seeds exceeds the concentration that causes shoot or root growth. Drying the germinating barley seeds at 50 degrees Celsius is one method to stop malting. Explain how this method would stop malting. So basically, the enzymes are denatured at higher temperature, like 50 degrees Celsius. At high temperature, the tertiary structure of the amylase changes. As a result, the active site changes shape. The active site then no longer complementary to the starch. High temperature dries the embryo, so the gibberellin synthesis also stops. Part 2. Suggest why malting is stopped before shoot or root growth occurs. So, to stop the sugar from being used, all right, and enough sugar present for beer production, because the sugar would then get used for the shoot and root growth. 4a. Genome-wide association studies find links between single nucleotide polymorphisms (SNPs) and phenotypic features such as human disease. SNPs are joint points on the DNA that vary in the population because of DNA-based substitution. A genome-wide association-wide study investigates the effect of genetic variation on a disease. A large number of people with the disease and a large number of healthy control individuals provide DNA. Microarray chips are used to identify the genotype of each individual at many SNPs. The Wellcome Trust Case Control Consortium study was an important genome wide association study. The study used a microarray chip that identified each person's genotype at 500,000 different SNPs. The study looked for links between SNPs and 7 different diseases. For each disease, 2,000 people with disease were tested. Their results were compared with the results of 3,000 healthy control individuals. Outline how microarray are used in the analysis of genome. Well, this particular question starts with a very big introduction. Not to get scared with this particular question, the main question is within our study limit. As you can see, outline microarrays and how it is used in the analysis of genomes. So first of all, we're going to say, you see, to study in order to study genomes in microarray, first of all, single-stranded DNA are obtained from the DNA. The DNA is then labeled with fluorescent tags. The microarray chip contains probes, and each probe is unique to a particular SNP. The DNA is then added, which hybridizes with the probe. Excess DNA is then washed up after hybridization. Then microarray chip is viewed under UV light and high-resolution digital camera. So the high-resolution digital camera, what it does is that it takes photo of it, and the fluorescence that it produces, which indicates the presence of SNP. Part 2. Explain why bioinformatics was important to study WTCCC study. So we know that bioinformatics was important because a large database is needed for this kind of study. The database is used to find SNPs and match fast and accurate with people, those that have SNPs. Computer software and algorithms are needed for statistical analysis like this. Figure 4.1 summarizes results from 3 diseases in the WTCCC study, the 22 human autosomes and the X chromosome. Chromosomes 23 are shown. Chromosome location with SNPs that are associated with the disease at a statistically significant level greater than 5 arbitrary units are shown in black. So, yes, we can see at number 1. Alright, number 6, and then type 2 diabetes, type 1 diabetes, rheumatoid arthritis. Alright, that's 6, we can see, find this value significant. Now the question says, identify the chromosome that contains SNP that have a high level of association with both rheumatoid arthritis and type 1 diabetes. So in type 1 diabetes and rheumatoid arthritis, we can see in both of them we can get this particular chromosomes SNP 6, and 1 is also very relevant. Alright, there is no other relevant, so 1 and 6. Now, with reference to figure 4.1, compare the genetic basis of the 3 diseases. Alright, so if we look into these particular diseases, rheumatoid arthritis and type 1 diabetes, nothing we can see on the X chromosome though. We don't see any level of association in terms of arbitrary units in X chromosome. So all diseases are autosomal and not sex linked. All diseases are associated with SNP on chromosome 6. As we can see on chromosome 6 there is SNPs that matches in all of the diseases. Type 1 and type 2 diabetes. Type 1 and type 2 diabetes SNPs are associated with chromosomes 12 and 16. As we can see, there is 12 where there is a match, all right, and then there is a 16 where there is a match. So we'll locate them. As you can see, 10 did not have any match with type 1 diabetes. So all right, then type 2 diabetes SNP associated with chromosome 10, but type 1 diabetes and rheumatoid arthritis does not have it. Arthritis and type 1 diabetes have higher level of association with each other than with type 2 diabetes. Rheumatoid arthritis has fewest number of SNPs. As you can see, rheumatoid arthritis has only 1 and 6. Point C. Individuals can choose to have their DNA analyzed on a microarray chip to predict their risk of developing different diseases. Outline the social and ethical considerations of the DNA analysis. DNA analysis is not available for everyone. However, it allows people to plan and organize care. Lifestyle change, example, losing body weight, take exercise as a part of their lifestyle. People can then prepare for early treatment as well. People may decide whether to have children or not to have children. Results may cause anxiety and stress. DNA analysis result may affect ability to get insurance, and also predictions may not be always accurate. Biodiversity can be considered at three different levels. Figure 5.1, 5.2, and 5.3 are images that shows three different levels of biodiversity. Describe the level of biodiversity most clearly visible in each image. In 5.1, we can see many different species available. In figure 5.2, we can see a variation within that particular genetics that is available within the same species. And in figure 5.3, we can see a particular habitat or an ecosystem. So figure 5.1 species, figure 5.2 genetics, and figure 5.3 habitat slash ecosystem. Part B. Prairie strips are restored habitat areas planted with native grasses and wildflowers. Prairie strips are usually located between or around the edges of fields of maize or soybean. Students in North America wanted to investigate biodiversity along a prairie strip. Outline how the student could measure the biodiversity of plants and insects along a prairie strip. So basically plants. The student can use quadrats along along line transect. The student then can estimate the percentage cover of each species from the particular quadrant. For insect, the student can use nets for flying insects in order to get estimate and use mark release recapture technique. The student then can use images of known species to use as identification keys. Part 2. Mathematical methods and statistical tests help to summarize and compare biodiversity data. Name the mathematical method or statistical test that the students can use to estimate the total biodiversity of a prairie strip. So, to estimate the total biodiversity of a prairie strip, Simpson's index of biodiversity can be carried out. To test whether plant biodiversity is associated with insect biodiversity, Pearson's linear correlation coefficient can be carried out. The prairie strips have environmental and economic effects. Scientists have made long-term measurements of the environmental effects of prairie strips. The scientists found that when prairie strips form 10% of a crop field, soil erosion decreased by 95%, mineral loss from the field decreased by 90% for phosphorus compounds, and mineral loss from the field decreased by 85% for nitrogen compounds. Crop plants need phosphorus and nitrogen to grow. Evaluate the economic effects of prairie strips on farming. So basically, prairie strips decreases crop area, which can lower the yield. However, prairie strips can have insects which may also feed on the crop, so less profit can be made in cultivation. Prairie strips makes the soil more fertile, so there will be higher yield even with a smaller amount of land. And less phosphorus and nitrogen compounds are needed to grow crops because they leach out from the soil less. This results in higher profit from smaller cultivated land. Since we are asked to evaluate the economic effects of prairie strips, so we have to write both for and against. So we have 3 points for both for and against. You can just choose any of the points that you feel are more suitable for your answering technique, and then you can answer from there. So guys, that's all for today's video. We're gonna start from question number 6 to question number 10 in our next video. Alright, subscribe to our channel to get a notification of videos whenever they're uploaded, and comment in your video which question paper that you want us to solve. Please make sure that they are from chemistry and biology, and subscribe to our channel and refer our channel to other people, those who will be helped by us. Thank you. Bye bye.